Hello, molecules and mischief makers. Your local science side group here, and today we are going to decode a topic called as cloning and selection. In that, we'll be focusing on how to stimulate plating efficiency, and also look at some of the ways in which we can, do suspension cloning. So, without further ado, let's get into the topic. So, most of the information which I'll be explaining in today's video is from a textbook, that is Animal Cell Culture by Ian Freshley. And yeah, so today will be only focusing on two topics, that is stimulation of plating efficiency and also uh, some of the methods. So these are one of the method for cloning, which is called as dilution cloning. So we'll be mostly focusing on this topic, that is stimulation of plating efficiency. So when cells are plated in low densities, you see that when you plate like less amount of cells, then the main problem what you see is that the survival rate is less. Like if all the cells die, then everything is dead. So but then like only in few cell lines. But then if you use like a continuous cell line, then also the plating efficiency drops by 10%. And then if you're using primary cell lines, it is 10%. For continuous cell line, there's no much problem because you're continuously changing the nutrient medium. And if you're using infinite or finite cell lines, the plating efficiency is very low, that is 0.5 to 5%. So there have been like many attempts to improve the plating efficiencies, even at low density, like for example, by increasing the nutrition and all that. One of the main problems we have is that cells, they undergo leakage through the diffusible signals, and because of that, and if they're diluted also, the amount of nutrition is less. So because of this, we are using a method. So if, for example, the metabolic pool of the leaky cell density, it will usually go into equilibrium with the surrounding medium. But if you take that cell and keep it in an isolated way, then this will not happen. So this is the same principle which is followed in capillary technique, which was developed by Stamford and his team in the year 1948. So this was mainly done for L929 clone of L cells. They were the ones which were first produced. So what we're doing is we're trying to make a confined environment using a capillary tube, so that there is enough nutrition, that in that local area you have enriched environment which can mimic high cell concentration. So that is one way in which we can overcome this problem. The other way is by using micro drop technique. These were developed later. So the capillary technique was developed first, and then we have the micro drop technique. Here what we're using is we are using liquid paraffin to create a high cell density concentration by keeping one colony separate from the other. And also this helps in easy isolation, or it becomes easier to isolate later. And along with that, over time, we know that the media improved, and because of the improvement in the media, we also see that the plating efficiency have improved over time. Park and Marcus in the year 1955, they were able to show that cloning cells, even by simple diffusion, along with using like feeder layers, like to give extra nutrition to the medium, of irradiated mouse cells, fibroblast cells, gave a very good cloning. But after that, the thing is you had to do like trypsinization in order to like separate the different colonies after they they grew. So now let us look into some of the conditions in which or how we can improve cloning efficiencies. So these are some of the conditions. The first one is medium and serum, hormones, carbon dioxide, intermediary metabolites, and treatment of substrate and using trypsin. So we'll look into some of the examples, like what type of medium and what are the different types of hormones that can be used to improve the clonal growth. So Now, let's go into the next one. So hormones, we kind of discussed the hormones. One is insulin. You can use dextromethasone, and some of the synthetic hydrocortisone can be used to increase plating efficiency for chick, myoblast, human glioma, fibroblast, melanoma, but they have to be removed 5 days before plating. Now coming to the next one, that is intermediary metabolites. So coming to intermediary metabolites, so one of the main intermediary metabolites that can be used are oxo acids. So oxo acids were previously called as keto acids initially. So oxo acids or keto acids can be used. In oxo acids, the common example is pyruvate. Pyruvate is an oxoacid which can be used as an intermediary metabolite. And another example includes alpha oxoglutarate, which was also initially called as alpha ketoglutarate, alpha oxo or alpha ketoglutarate. And another intermediary metabolite is nucleosides. So some of the nucleosides can be used in order to improve the plating efficiency, and we can also supplement the medium. So medium can be supplemented. So along with that, so pyruvate. So this pyruvate, it is usually added to this medium, that is which most of you might be familiar with. That is the DMEM medium. So if you add pyruvate to the DMEM medium, it is said to have or stimulate cloning efficiency. Uh, so that is Dubelsko's modification of Eagle medium. So these are some of the intermediary filaments which can be used, like oxoacids, which is previously called ketoacids, pyruvate, alpha oxoglutarate, also called as alpha ketoglutarate, nucleosides, and also supplement the media supplements. And pyruvate is usually added to the DMEM medium. Now let's go to the next thing, that is carbon dioxide. So how much carbon dioxide is ideal for stimulating the plating efficiency? So from experimentation, they found usually we see that in most of the cloning experiments, 5%. So 5% carbon dioxide is usually used, but it is also known that 2% works well. For many cell types, 2% carbon dioxide works better, especially if you're talking about human glial cells. So human glial cells, and also when you're talking about fibroblast cells. So fibroblast cells and human glial cells, you see that carbon dioxide works, and along with that you can use HEPES, HEPES, which is about 20 micromolar, and pH adjusted to 7.4. So this is about carbon dioxide and some of the conditions which can be used. 5% usually used, 2% works better for many of the cell types, especially for human glial cells and also fibroblast and HEPS. And the pH is maintained to 7.4. And then let's look into the treatment of substrates. So what are the other substrates that can be added in order to improve cloning efficiency? So in that we have polylysine. So polylysine polylysine can be used. So polylysine is mostly used in order to improve plating efficiency of human fibroblast cells. For human fibroblast cells, the polylysine works better. And even in low serum concentration, you can use polylysine. As you know, serum is very expensive, and also from the ethical perspective, if you look, it's always better to use medium with low serum. So if you have a low serum concentrate of the medium, then we can use polylysine in human fibroblast cells. So the amount of polylysine is also calculated, that is about 1 mg per ml of poly-D-lysine is added to the plates. So if the plate is about if the plate contains about 5 ml, or if it is a 25 centimeter cube petri plate, centimeter square petri plate, so or 5 ml, to that you'll be adding about 1 mg per ml of poly D lysine is added. And then you have to wash the plates. So you will be washing the plates using 5 ml of PBSA per 25 centimeter cube plate. So 5 ml PBSA is used, and then the plates are used, like they are used for culturing. So that is treatment of substrate. So under the treatment of substrate, you can use polylysine to improve the plating efficiency of human fibroblast cells, which are grown in low serum concentrate. For that, we'll be adding about 1 mg per ml of poly D lysine to the plates, which are about 5 ml of 25 centimeter cube. You have to remove the plates, wash it with 5 ml of PBS and per 25 centimeter square plates, and then they're used for culturing. So that is about the treatment of substrate. Now coming to the last thing, let's talk a little bit about trypsin. So how much trypsin can be used to improve the plating efficiency. So in that, if you see, so purified trypsin is used. So in trypsin, we are using purified trypsin, which is about 0.05 micrograms per ml. And this is if you are using purified one. So purified one, you'll be using this much. If it is crystallized, that is, if the trypsin is twice crystallized form, if you're using that type of trypsin, twice crystallized purified one. So that which is usually maintained in 4 degrees Celsius. So then you have to also recombine it with uh, for or you can use different types of trypsin. One is if you're using purified trypsin, you have to use 0.05 μg per ml. If you're using a twice crystallized trypsin, usually it is maintained at 4 degrees Celsius, and then you also have recombinant trypsins. So under the recombinant trypsin, one of the example is trizine. So trizine, it is available from the Sigma company. Trizine is a recombinant trypsin which can be used. Or you can also use another recombinant trypsin, that is tri-LE. So these are some of the examples of trypsin which is used in cloning, like in the last steps of cloning where you want to isolate the single cell cultures, you'll be using trypsin. And these are some of the examples of trypsin which can be used. If you're using purified, it is 0.05 um, micrograms per ml, or if you're using twice crystallized purified trypsin, which is maintained at 4 degrees, or you can use recombinant trypsin like trizine from the Sigma company or tri-LE. So that is all about. So with this, we have actually completed uh, everything about what are the different ways in which you can stimulate cloning efficiency. So we talked about serum, hormones, media, intermediary metabolites, carbon dioxide, and also some of the treatment of substrates and trypsin, which can be used to improve cloning efficiency. So now let's go into the next topic, that is suspension cloning. Suspension cloning is also like a type of cloning method. For example, in some cells, like particularly you have hematopoietic stem cells or virally transferred fibroblasts, the clone, you can easily get the clones in a suspension rather than in a solid medium. So to hold the colony together and prevent a mixing, so you cannot, like the suspension, it's in a liquid form, right? So it keeps moving. So in order to prevent that, we'll be using another layer of agar, or we can also use a methoxyl is plated on the agar like an underlay under the dishes, so that it is like, it's easy for you to take the clones out. So we'll be looking at how can you make this agar and how the methoxyl is made for suspension cloning. So first, let us look into the suspension cloning method so this is mainly done for cultured cells, primary suspension from bone marrow or tumor cells. So these cells you can see in the diagram. I hope the diagram is clear. So there are like three ways. So the first step is preparation of the agar underlay. The second step is preparation of the agar medium, and then the third step is preparation of seeding of the cells. So first, what you have to do is you are taking the cultured cells and they're suspended in a agar. So you can either suspend them in agar, or there is another type of agar called as a low melting temperature agar, which is allowed to gel, and the colonies are formed in the suspension. So if you're using this as an underlay, like you are using this as a base, like so that on top of that the colonies can grow. Using an underlay prevent. So why we are using this is mainly to prevent the cells attaching to the base of the dish. So this like adds like a buffer layer on top. So for that you can use agar or low melted agar suspension can be used here. So the first step is preparation of the agar underlay. So for this, you will be using, so the amount of agar is used here. You can see it is 1.2% agar is prepared using ultra pure water, uh, and the temperature at which it is kept is 55 degrees Celsius, and it is mixed with 2x medium at 37 degree Celsius to give uh, the required amount percentage, that is to give this much. So to give 0.3% of the agar which is required for cloning. So what you'll be doing, you'll be preparing the first step is to prepare this one. The first step is to prepare the agar underlay. For that we need 1.2%. Initially we start with 1.2% of agar in ultra pure water, which is maintained at 55 degrees Celsius, and then it's mixed with 2x medium to give 0.3% agar. That's the final concentration of the agar. And the use of low melting point agar allows all the solutions to be maintained at 37 degrees Celsius. But a problem with this low melting point agar is that it's easy to maintain the temperature. Like in 37 itself, you can maintain them. It will not harden so much. But the problem is that it will take a long time to gel. So that's a problem. Now coming to the third step. So you have prepared the agar medium, like you have made different concentrations. The final concentration is 0.3% of agar is made. And then you'll come to the next step, that is preparation and seeding of the cells. So these cells, so you can see the cells are grown in the suspension which is derived from the bone marrow and trypsinized from attached monolayer, and these are counted, diluted, serially diluted, and the final product is diluted with agar and agarose, and it is seeded. You can see the cells are seeded onto the agar underlay. So this is how cloning and suspension is done using agar as an underlay. So agarose as a reduced component. So some of the uses, so agarose as a reduced component sulfate polysaccharides which can be substituted by agar, and some of the types of agarose like low gelling temperatures can be easily obtained, and it can be returned to 37 degrees Celsius. And because of this complexity, the main problem when we're using agar is the temperature. So you have to be very careful while and keeping the temperature because they get hardened soon. So most of the laboratories, they prefer another method which is called as methoxyl, which is a viscous liquid. It is not a gel. Instead, it's a viscous solution. So this is developed by them, Buke and team, in 1979. So it has a very high viscosity when it is warm, but it is not a solution and it is not a gel, and it will sediment slowly. Therefore, you have to use it as an underlay. Methoxyl is used as an underlay, and the colonies will form an interface between the methoxyl as well as the agar, uh, which and also compared to agar, since this is a viscous liquid, the photography or getting the results is much better if you're using methoxyl as the So this is the protocol. I mean, you can just go through the protocol. I'll be not going into the details for cloning in the agar, all the technical details like how you have to make the medium and the amount, like for example, how much grams of agar, like 1.2 grams of agar has to be added using the ultra pure water, and then transfer them in water bath and so on. So this is a protocol which you follow. It's also in the textbook. You can go through it for your reference when you're cloning in agar. So these are the materials which is required, and mainly you'll be making that concentration. You'll first make the underlay, and once you make the underlay, then after making the underlay, you have to make a cell suspension, and it is put on top of the underlay, and then it is kept like it, the cells are allowed to grow. Like you have 0.3% agar, it is kept at 37 degrees Celsius by diluting, and then you have to prepare different dilutions, and then it is added, and finally you'll keep the cells for growing, and you can suspension. So suspension cloning is done. So now the other type of cloning, which is little better than agar. The main problem with agar is that the temperature has to be maintained, and it's little difficult to take the final results. So we use this cloning in methoxyl. So here what you have to do is that you are first taking diluting the cells from 100,000 to and 3,700 cells, depending on the expected plating deficiency. So you will be diluting it so that you can get better cloning, because enough nutrition will be there. And then here you have the 2x medium plus 1.6% methoxyl is added, and so when you add this, you'll get around 0.8% methoxyl and 1x medium. So that is added. So dilution is 1:100 times dilution is done using this 0.8% methoxyl, and then it is seeded. So here again, you're also it's like you're combining both methoxyl. So it doesn't mean that you're eliminating agar in this method. So there is an agar underlay, that is in the other one we use 0.6% of 0.3% of agar. So here there is a little more. So 0.6% agar or agarose underlay is there. And on top of that, you're also using methoxyl, because it's a viscous liquid, to keep the so that you can see the colonies better, and also I mean, you don't have to worry so much about the temperature. So this is the protocol for cloning using methoxyl. So you can go into the details. So you can just gives you how exactly you have to do the dilutions and perform the experiments. After all that, you are incubating the dishes in a humid incubator until the colonies are formed. And you can also see there's an interface between the agar and the methoxyl, and a fresh medium can be added. So once the medium gets exhausted, like 1 ml per dish or well is added. Like after 1 week, you have to remove the medium and replace it with fresh medium. And after uh, fresh medium, and after 2 weeks, without disturbing the colonies, you have to add new medium, so that the cells are able to grow and there is enough nutrition for them to grow. This is Science Side Gov signing off. If this video even sparked one of your neuron, please consider liking, sharing, and subscribing this video. Let's keep culturing curiosity. Until next time, I'll see you in the next one. Thank you so much.